Well, it's always very difficult to start a talk when your time is up, but I'll try to do my best to go fast. So first, I'm very honored and very pleased to be here, Philippe. And so we know each other since quite a while now. So I was not part of your lab, but I didn't have this privilege. But I was not from the École Polytechnique, so that's a really bad start. But anyway, we have been collaborating, and you are not either, right? Okay, so we have been collaborating on the key molecules that class I think is going to talk about. And it was a pleasure, and I should say that I have been always impressed, and I'm still impressed by all the talents that you have. I mean, it's really incredible from science and discoveries in science, which is not that frequent, and also to the management of science in France and in Singapore, and also to the writing of books, and even philosophical books, and so like Altruity, as you've been referring to. So it's really impressive, and I should say that you are a true example for all of us here, for your passions of science in particular. So thanks for being what you are. So what I would like to present to you today is really relates on innate immunity. And I would like to focus on a peculiar subset of lymphocytes, which are called natural killer cells, and which belong by some aspects of their biology to this class of innate immune cells. So what we are trying to do in the lab is actually to combine different kind of approaches in the mouse and in the human model, trying to decipher the mechanism by which NK cells do what they do, and what they do is actually to kill cells. They are cytotoxic cells, but they can also produce a vast array of cytokines and chemokines, as I will tell you in a minute. Indeed, NK cells have this propensity, which is very fascinating to us, to recognize stress cells from healthy cells. And they do that using a variety of cell surface receptors. And I will touch on the basis of this recognition later during the talk. But in short, they can recognize a vast array of cells that I will try to call stress cells. And the stress can be induced by a very large array of conditions that can include tumor transformation, microbial infection, but also physical stress and chemical stress. And in the presence or in the absence of antibody, NK cells can recognize these cells. And as I've been telling you, they can lyse these cells, but they can also produce cytokines. And the combination of that makes that NK cells are not effector cells and participating directly to the elimination of stress cells, but they can also participate to the shaping of the adaptive immune response, as is seen here. As a consequence of that, NK cells are involved in many different processes in vivo. They have been shown in human and in the mouse to be involved in the elimination of tumor cells. This can be manipulated in particular in the context of hematopoietic transplantation, and this is very important at present. They are also controlling some kind of virus infection, some specific class of virus infection, in particular herpes virus infection. So it's true that it's more obvious in the mouse than in humans, but with the studies that Jean-Laurent has conducted recently, in particular in the cohort of Irish travelers, it turns out that it might well be true also in humans. And very interestingly, and I think it's still an enigma, but NK cells are probably involved in reproduction. And this is something which has been ignored by most of us, but it's true that NK cells literally invade the uterus, in particular the pregnant uterus, and they might contribute to the remodeling of the arteries during pregnancy, which is very important for the fetus development. I should say that an emerging theme in the field of NK cells is also detrimental roles in the context of inflammation, where they might actually contribute to tissue damage. So what we're trying to do in the lab is trying to understand how NK cells do what they do using genetics. So very inspired by the work of Chris Goodnow and Bruce Butler, we have been setting a very large genetic screen in the mouse using ENU mutagenesis. So in short, we are trying to reproduce what Jean-Laurent is doing using patients, but we are introducing the mutations ourselves. And in order to do that, we are using this chemical agent ENU, and ENU is very important for two reasons. First, ENU is introducing point mutations in the genome, and in particular, these point mutations are also present in the spermatozoids. So that now the male mouse that have been injected with ENU can transmit these point mutations in the progeny. And then you can ask the question that you want, in just a question of time and question of space, particular mouse house space. But you can actually try to track the genes which are involved in your function of interest. So in our lab, we are screening animals at the G3 level, trying to concentrate on recessive mutations that will be observed then in homozygous, in animals carrying homozygous mutation. So here is an example of what we have been doing. So we have been setting up a large array of NK cell-based screens, in vivo and in vitro. And this is an example of what one can see in the different mini pedigrees that we obtained in our zoo of mutant mice. And so as you can see here, definitely in this mini pedigree, as well as this one and this one, you have a fraction of mice in which NK cells are outliers, as you can see here, looking at the propensity of NK cells to actually kill some target cells. So far we have been screening more than 4,000 G3 mice, which is quite of a work, as you can imagine. And I would like to tell you that although the yield in terms of the number of mice that are finally of interest for us is quite low, because I should say that we have probably kept less than 10 mini pedigrees, it was very interesting for us because it allowed us to actually pinpoint some very important checkpoint in NK cell differentiation. This is here a scheme that recapitulates NK cell differentiation in the mouse, and you can see that with those names here, which are the names of the mutants that we have generated, one could pinpoint, again, very important steps in NK cell differentiation, and I would like to focus on two of these mutant mice today. And the first mouse is called Genista, and you will see that in those mice, actually NK cells are hyper-responsive. And this is the work of a very talented PhD student who's actually looking for a postdoc. His name is Baptiste Jäger. So this slide is to illustrate how hyper-responsive are the NK cells when isolated from the Genista background. So here you have the capacity of NK cells to produce gamma interferon when cultured in vitro for 4 hours in the presence of YAC tumor cells or cross-linking antibodies. And you can see that, in fact, indeed, when NK cells are coming from the Genista background, they're hyper-responsive. So one of the first questions that we ask at this occasion is actually to find out whether the phenotype is intrinsic or extrinsic. And so one way of doing that is actually to isolate wild-type NK cells and inject those mature wild-type NK cells in a Genista mouse and to analyze the NK cells. And as you can see here, wild-type NK cells become hyper-responsive when transferred into Genista mice, showing that at least one part of the hyperactivity of NK cells observed in Genista mice is actually due to an NK cell extrinsic factor. One can obviously perform the reciprocal experiment when one injects Genista NK cells into a wild-type mouse, and sure enough, as you can see here, so here the donors are Genista background. When the Genista NK cells are transferred into a wild-type host, they regain reactivity, showing again that some of the factors that are involved in NK cell hyperactivity are actually NK cell extrinsic. And then I would like to illustrate the advantage of working as a CIML in Marseille. There are numerous advantages of working with us, as you probably know, but one of the advantages is that we are probably the last communist after Cuba. Meaning that we are trying to share everything that we can. And in particular, for this huge program of genetic screening in the mouse, we were collaborating with Bernard and Marie Malissen to set up this zoo of ENU mutant. And it turned out that actually our PhD student was almost fighting with a postdoc from Bernard and Marie, chasing after the same mice. And okay, it really didn't make sense to us because they were not supposed to work on NK cells, and actually they did not. They were chasing for another phenotype, which is the one illustrated here. Those genista mice are actually lacking neutrophils. And this is true in the blood, this is true in the spleen, and this is true in the bone marrow. So actually there is a strong effect in one of the transcription factors, which is very important for the differentiation of neutrophils, which is called GFI1. So, as you probably know, neutrophils are very short-lived, and they are also very important. One cannot live without neutrophils. They are very important to control a number of microbial infections, in particular bacterial infection. As you can see here, a neutrophil interacting with bacteria. But because of this short half-life, they really are presented as effector cells, and that's it, with not much of immunoregulatory function. So because we were chasing for an NK cell extrinsic factor, and because those genista mice were actually lacking neutrophils, we asked a very simple question, whether actually the lack of neutrophils was not responsible for the hyper-responsiveness of NK cells. So in order to tackle this question directly, we've been using antibody depletion in vivo using two kinds of antibodies. So as you can see, there is a nice depletion of neutrophils, although it's not complete. And I should say that the extent of depletion is much more important in the genetic mutant. But despite this caveat, one can see that using these two different kinds of antibodies, one can restore the hyperactivity of NK cells when one depletes neutrophils in vivo, showing that, in fact, we might have been true in saying that neutrophils were indeed the extrinsic factor in the genista mice, which was responsible for NK cell hyperactivity. But as I've been telling you at first, we are trying to combine human and mouse studies. So we turned to human to find out whether that was true and conserve across species. So in collaboration with Jean Donadieu, who is actually involved in the French registry of neutropenia, we've been able to harvest quite important, considering the frequency of those patients, of samples from patients presenting neutropenia. And two kinds of neutropenia, severe congenital neutropenia, SCN, which for most of the patients is actually due to a mutation in the gene called elastase2, and then autoimmune neutropenia patients, which are neutropenic for a completely different reason. They have circulating antibody that depletes neutrophils, and they still have neutrophils in the bone marrow, but they don't have circulating neutrophils. But it was very interesting for us to see that in both conditions, which somehow recapitulate both the antibody depletion in the mice and the genetic mutation in genista, we also see hyperactivity of NK cells in those neutropenic patients. So we do think that these results, which have been recently published, actually highlight a new crosstalk within the innate immune system between neutrophils and NK cells, and I was really thrilled by the presentation of Jean-Laurent Casanova, not only because it's always outstanding, but also because of the new cytokines that you presented, Jean-Laurent, which really might be one of the elements that makes that neutrophils help in the reactivity of NK cells in vivo in both human and mouse. And now we'd like to switch to another kind of mouse that we have been able to generate, and this is a super mouse. As you can see here, this is the work of a very talented postdoc in our lab, Emilie Narni-Mancinelli, and we have called those mice Noé. And we call this super mouse because actually the NK cells in these Noé mice are hyper-responsive, just the other way around, the genista mice. And one can see that in different kind of experimental conditions. First, in vitro, when we generate lac cells and stimulate those lac cells with YAC tumor cells, one can see that NK cells from a Noé background are actually hyper-responsive in terms of gamma secretion as well as degranulation. And this is also true when one stimulates freshly isolated NK cells using cross-linking antibodies against NK1.1, NKp46, as well as a combination of stimulation of IL-12 and IL-18. But so obviously we wanted to challenge our mice in vivo to really find out whether this hyperreactivity of NK cells could be important in other conditions. And as probably some of you know, one of the best models to look for the function of NK cells in vivo is to infect them with herpes viruses, in particular the mouse cytomegalovirus, MCMV, because it has been shown by several groups that, in fact, NK cells are key to the control of MCMV in VAC6 mice. The thing is that when one uses huge doses of viruses, as we have been doing in this particular experiment, despite the presence of NK cells in wild-type mice, all the mice would die in 8 days, as you can see here. So the obvious question was, the obvious experiment to do was to challenge no mice using the same dose of virus, and it was really interesting to us because no mice were dying in these conditions of infection, and hence NK cells, well, it suggested that in fact the NK cell hyperresponsiveness could be involved in a very drastic and impressive resistance to MCMV infection. So to address this question directly, we've been depleting NK cells from the NOE mice, and as you can see now, there is no resistance of the NOE mice to MCMV anymore, showing that in fact this phenotype is NK cell dependent. So this is associated with a number of other parameters, but in the interest of time, I'm not going to show you the data. You really have to believe me. This is associated with a reduced viral load. There is also, for the NK cell aficionados, a similar frequency of LY49H NK cells, which is a subset that directly recognize MCMV-infected targets. Is also associated with increased NK cell response in infected NOE mice without restimulation in vitro. So it was really interesting, but it was even more interesting when we actually have challenged the mice using other kinds of viruses. This is one of the examples. Here is the flu, H1N1, and as you can see here, there is a drastic resistance to NOE mice to this other kind of virus infection. So now we had to identify the mutation responsible for the NOE phenotype. So as Jean-Laurent already said, nowadays genome resequencing or exome sequencing is the way to go. So this is what we have done using an Illumina platform in Barcelona. And we knew, and I had no time to show you that the mutation was actually NK cell intrinsic. So we were looking for homozygous mutation, and we were looking for NK cell intrinsic phenotype, which was obviously very important for us to decrease the number of gene candidates. And we ended up with 4 gene candidates. And one of them was really interesting to us because it was NCR1. So what is NCR1? Well, NCR1 encodes for a cell surface protein called NKp46. And NKp46 is really a key molecule in the NK cell field because it's undoubtedly the best marker to look at NK cells across mammalian species. It's also mostly expressed on NK cells, and it's highly conserved, both at the protein level, but also at the regulatory region of the genes themselves from opossum to human. So it's really something which is a feature of NK cells. And the NOE mutation was actually a point mutation in this first domain. And as you can see here, this mutation prevents the cell surface expression of NKp46. Here we are getting an NK cell, CD3 minus, NK1.1 plus. In wild-type situation, they express NKp46. In the NOE mice, they still express NK1.1. They're still CD3 minus, but they don't express NKp46 anymore. And we have shown using transfectants this is really the mutation which is associated with the lack of cell surface export. But as you know, the problem with the INU mutation is that you don't induce only one mutation, but several mutations, actually about 30 mutations in G3 mice. And so we had to show, using genetic tools, that it was really the mutation in NKp46 which was responsible for the phenotype that we were interested in. So in order to do that, we have crossed the NOE mice to transgenic mice expressing the human NKp46. So we had generated those mice years ago, and so those are not back transgenics, but still they do harbor a 240 kilobase human DNA sequence that encompasses the human NKp46 gene. Hence, these human NKp46 transgenic mice actually have all the regulatory regions of the NKp46 gene, and as a consequence of that, the pattern of expression of human NKp46 in the transgenic mice mimics exactly the pattern of expression of the endogenous NKp46 in wild-type situation. So it was very interesting for us to get those mice because we could create a different kind of genotype, so the wild-type mice to begin with that express endogenous mouse NKp46, but then we could compare these wild-type mice to Noe mice, which do not express any mouse NKp46 anymore, and we could compare those mice to mice that now are on the Noe genotype, but express the human NKp46 transgene, and hence the human NKp46 protein in place of the mouse NKp46 protein. I should say at this stage that this is really important that the human NKp46 is highly homologous to mouse NKp46, and when I say highly homologous, it's more than 95% identity at the amino acid level. And it was very nice to see that, in fact, this genetic rescue was actually restoring the NK cell phenotype in this in vitro test, as you can see here, but also restores the sensitivity of the Noe mice to normal. So proving by this genetic means that, in fact, the mutation in NKp46 was responsible for the Noe phenotype. But now the problem is that NKp46 has been described for 15 years by the Moretas, who actually identify this protein first in human, that NKp46 is an activating receptor. We still don't know what the ligands of NKp46 are, but I'll show you that there are some ligands which are constitutively expressed in the mouse. But irrespective of the nature of this ligand, when one through a number of experiments, one can show that, in fact, NKp46 is actually activating NK cells. So the question that we had is, how come the lack of a NK cell activating receptor is actually rendering the NK cells more reactive, which is kind of counterintuitive in a way. So there are three aspects, three sets of data that you should see before trying to understand the reason why this is happening the way that it does. First, we have been using a large transcriptomic approach to find out what could be the mechanism behind this hyperactivity. And to make a long story short, we have seen that the pattern of expression of the transcription factor Helios was actually mimicking NK cell reactivity in the three type of mice that I've been presenting to you, wild type, Noe, and Noe rescued by the human transgenic. So it turns out that Helios is an interesting transcription factor because the transgenic expression of Helios in B cells is actually also leading to hyperresponsiveness in a way that mimics what happens for the expression of Helios in Noe NK cells. But we had to show that it was not only associated, that it was causative of the hyperactivity of NK cells. So we have been silencing through shRNA approach Helios in Noe NK cells, and as you can see, the silencing of Helios is actually restoring the reactivity of NK cells to wild-type levels. So I think that it shows that Helios is really important in the hyperactivity, which is downstream of the engagement of the lack of engagement of NKp46. Second set of data, which is important, there is an endogenous ligand for NKp46. It's too preliminary to tell you about the nature of this ligand, but as you can see here, when one culture wild-type NK cells with CD11C plus splenic cells, one sees an activation of NK cells by looking at gamma interferon secretion, GM-CSF secretion, TGF-beta secretion, and this is inhibited by blocking antibodies against NKp46. And third set of data, this is actually not a set of data, but rather a recapitulation slide. This is the model that we are coming up with. We are actually thinking that during the development of NK cells, the cell surface expression of NKp46 allows NK cells to detect the presence of endogenous NKp46 ligand. And through an activation of NK cells that leads to the downregulation of Helios, that actually downregulates NK cell reactivity. So hence, in the absence of NKp46, this downregulation of Helios is not at work anymore, explaining the hyperreactivity of NK cells. And I think that this is a possible illustration of a very important thing in the field of innate immunity and NK cells in particular, is the fact that there are some adaptive aspects of innate immunity. And I would like to propose that NK cells are actually tuned by their environment. There is nothing like an innate immune response which is not tuned and adapted to the host. So this is an example of that. NK cells are equipped by two kinds of receptors, activating receptors, such as NKp46, inhibitory molecules that Claes Scherer is going to talk to you about, in particular inhibitory molecules that recognize the MHC class I molecules, and this sensing of the host through this activating receptor and inhibitory molecule is actually calibrating the effectiveness, the efficiency, the reactivity of NK cells in vivo. And so in the absence of this engagement of NKp46, lack of engagement of activating receptor, there is NK cell hyperreactivity, and as Claes will tell you, in the absence of this engagement of inhibitory molecule, there are actually hyperreactivity. We think that what's going on here is actually desensitization. It's just a very common biological process. When NK cells are receiving too much of activation, they are desensitized, and we favor this hypothesis to explain that the lack of inhibitory molecule is actually driving NK cells into an hyperreactive state. And a reciprocal kind of argument could be proposed to explain the hyperreactivity through NKp46 and Helios in our Noe mice. So now the question that we can ask also is, what are the advantages of having NKp46, which is completely conserved through evolution, as if you don't have any NKp46 anymore, the mice are resistant to viral infection, and NK cells are actually doing better. But as I will show you, there is a cost for that, as always in biology. And the cost is that if you have too much of NK cells, so to speak, when you have boosted NK cells, actually the cost is on the T cell side. Meaning that in different models, and again, in the interest of time, I have no time to go through all the data, but when you look at the T cell response, and here we've been looking at the CD8 specific, antigen specific response. This is true also for CD4 T cells. It's true also for the memory T cell response. The fact that you have boosted NK cell response is actually decreasing the efficacy of the T cell response afterwards. So basically, this is recapitulated in this cartoon. When you have a high dose of pathogen, nowhere resistant, and is likely good for the host, obviously, but a lowest dose of pathogen, you have really no difference between wild type and Noe mice, but the big difference is actually you have a weaker T cell response. So it's probably important to boost innate immunity, but as usual in biology, it's not a curve like that, but it's rather this kind of Gaussian curve, and the thing is to optimize how to boost innate immunity in different conditions. So NK cells are hyper-responsive, well, the NK cell hyper-responsiveness, is dampening T cell response. So, and I will finish by telling you that despite this cost, there are clinical conditions in which it might be very important actually to boost NK cell reactivity, in particular when T cells are not there. And this is the case in hematopoietic transplantation, where actually NK cells are coming back before T cells, and patients are very susceptible to a variety of virus infections, in particular CMV. So it was very important for us to see whether one can manipulate the NK cell compartment through NKp46 to recapitulate the hyper-responsiveness that we have seen in the Noe background. And this is what we are doing with Innate Pharma, a biotech company which is specialized in the manipulation of innate immunity through antibodies. And this is indeed the case. When one stimulates, actually, or when one injects, I should say, blocking anti-NKp46 antibody in the mouse, one can see that one somehow recapitulates the hyper-responsiveness that we have seen in the Noe mice. This is very preliminary experiment, but at least it tells us that there might be a new means now to manipulate NK cell activity in vivo in patients. So to conclude, I would like to, I hope that I convince you that NKp46 and Helios regulate NK cell responsiveness, that upon infection with high dose of viruses, highly reactive NK cells can promote host survival. We are actually crossing the Noe mice to the RAG background now to see whether they are really doing that in isolation, which really might be the case. But the fact that NK cells are hyper-responsive is not always beneficial to the host, as hyper-responsive NK cells during T-cell priming phase can affect the generation of fully protective memory T-cells. And finally, we have preliminary experiments showing that one can modulate NK cell reactivity by injecting blocking anti-NKp46 in vivo, which is completely counterintuitive, but it does work. And this has been recently published. I would like to finish here by thanking all the people who have been involved in that work, so in particular the people in the lab. This program, as all the programs in the lab, actually co-lead with Sophie Golini. And this particular program on Dory mice was really the work of Emilie Nardi-Mancinelli in collaboration with these other people. As I've said, I would like to thank also the funding, in particular ERC, and also our collaborators, so Bernard and Marie, for many reasons, the people in Barcelona for the genome sequencing, Bruce, for his inspiration in setting up ENU at CIML, and also Innate Pharma on our collaborators for shRNA experiment. Thank you very much for your attention.